In this lecture, we will talk about DNA damage and repair. DNA damage can take place due to certain reasons. One is the physical mutagens, other is the chemical mutagens. Physical mutagens include non-ionizing radiation such as ultraviolet radiation, in which electrons are excited, but they do not leave the molecule and they may come back later on. So this ultraviolet radiation causes some change in the DNA, electronic transition in the DNA molecules, such as in nitrogenous bases, for example. In ionizing radiations, these ionizing radiations are stronger, and when they hit some molecule, some electrons, they are removed from that. So, such as X-rays are very strong radiation, and then beta radiations from some, in some cases, or it may be like some other type of radiations also, which can cause ionization of the molecules. Another type is chemical mutagens. They include different types of changes like alkylated bases, in which alkyl group is added to the nitrogenous bases, methylated bases, then there are pyrimidine dimers can be formed. All these lead to DNA mutation or DNA damage. Another type of DNA damage can take place during meiotic recombination. In case of meiosis, when there is a chiasma formation takes place, during this recombination process, there may be some errors that may arise out of that. Maybe single nucleotide or maybe more than one nucleotide change. Then there are proofreading activities, as I discussed in my lectures on DNA replication, that DNA polymerases have good proofreading activity in the 3' to 5' exonuclease activity. But this is in some cases, like in case of prokaryotes in E. coli, 10 is to power 5, 10 is to power 9 is the chance that after these much nucleotides, there is one mutation can take place. So in any case, there is a chance that proofreading could not take place, and DNA polymerase just skip that particular change, so that may lead to the mutation. So why we need to discuss this is we need to discuss because if this mutation left as such, it may lead to some lethal side effect or it may be lethal for the cell or even for the organism. You might have heard of many genetic diseases where only single nucleotide change can cause so much problem with the patient. Uh, and most of these mutations are only single nucleotide change, which are called as point mutations. This point mutation may be lethal or it may be silent. Lethal ones are those which are damaging for the cell or even sometimes for the organism. And silent mutations are those they don't have any effect on the on functionality of the particular protein which is being synthesized after transcription and translation. So here is one example of frame shift mutation. This is a sort of mutation where whole frame is shifted. Uh, it may be it may take place by addition or deletion of a nucleotide. Like here in this example, this AUG is for codes for methionine, similarly CCC codes for some amino acid, and similarly these UCG and all that. If we add a nucleotide here, you can see whole frame is shifted downwards. Uh, just for reminding you that these three nucleotides they combine to form a codon, which gives a message for a one amino acid in the during the translation. So this mutation you are seeing here is after the replication, then the transcription at the transcription level. But even you can give this at the DNA level. You just replace U with T, and but ultimately, of course, it is transcribed into RNA, and which is further protein is synthesized. So just by the addition of one nucleotide, you can see whole frame is shifted. So it means altogether different proteins will be synthesized. So that protein will not be required by the cell. And if we delete any of these nucleotides, like from this strand, you just delete any of these. For example, this C is deleted. Even then, the whole frame will be shifted. So this is a very significant part of this, that this frame shift mutation, how it takes place. And if the change in the DNA molecule is that purine is changed with pyrimidine or vice versa, in that situation there is more stressful condition in most of the cases, where purine is replaced by pyrimidine or vice versa. Then we have transition. Transition is also a single base nucleotide point mutation, but in this case purine is replaced by purine, or pyrimidine is replaced by pyrimidine. This is shown by pyrimidine, this is by the purine. So in this case alternate purine or pyrimidines they are used. So even then it depends uh, if there is some change in the amino acid, and that change, if it is in active part of some enzyme or some important part of some transport protein, etc., so that will cause problem for the cell. But nature has not left us in front of such type of DNA damage or mutations. Otherwise, life would not have been possible on this planet. So there are at least 4 different systems that exist in case of E. coli cells, just to counter these types of damages or potential mutations. So that's why it's very important to discuss and study that if such damage is not removed or such mutation is not removed by some method, that may be lethal for the cell, or it may cause problem for other organisms. So we will discuss all these 4 methods. These are minimum 4 methods. There may be other as well. So we will discuss one by one each of these. So first is the mismatch repair. As the name implies, mismatch means that this base pair is mismatched. For example, okay, first I will discuss how this process takes place during replication. There is a parent strand and a daughter strand. Here blue ones are the parent strands and this red one is the daughter strand. And the parent strands, these are methylated, as you can see CH through 3 groups at various places. And this replication goes on in this direction. At a certain period of time, soon after replication, we get this type of hemimethylated DNA, where one strand, the parent strand, is methylated and daughter strand is still unmethylated. So after that, this strand is also methylated and both strands are methylated. Why this is important? This is important to discriminate which strand is the parent and which is the daughter. So cellular machinery basically distinguishes between the parent and daughter strand by the methylation. But after some time, when both strands are methylated, those cannot be discriminated or distinguished. So this DAM methylases are the enzyme which actually methylate the daughter strand. Here you can see that the other strand is also methylated. Now, how this mismatch repair? What is mismatch? Mismatch is a mismatch of the in the base pair of the two strands. When the new strand is formed, the parent strand is used as a template, and new strand is a complementary copy of that template. So here, if there is uh, A here in this strand, what should it be? Here should be T. But if, for example, C is added, first DNA polymerase will use as proofread activity. But here we are talking about the situation where proofread activity uh, is skipped due to certain reasons. So what happens if a mismatch is there? You can see this is a mismatch. So uh, there is certain sort of machinery that binds over there, and these are the proteins. I think they may be. So there are two proteins, MUT and MUTS and MUTL. These proteins recognize this mismatch, and ATP is hydrolyzed, and this is called as MUTLS complex. And then there comes the MUTH. Again, ATP is hydrolyzed here, and this complex is formed where all these three proteins are there. Hydrolysis of ATP or ATP requirement makes this energy dependent process and also gives a certain specificity to the to this process. Now, uh, a few nucleotides away from this complex, where this complex is formed, meaning where the mismatch was, there is a cleavage that takes place. There is cleavage takes place here, like it's like phosphodiester linkage is missed. And now here comes the importance of methylation. So as you can see, we needed the cleavage in the daughter strand because mistake or mismatch, that error was due to wrong nucleotide in the daughter strand, not in the parent strand. So methylation helps here significantly to find out which strand is daughter and which is the parent strand. And what happens next? As you can see here, uh, there are two pathways here. It may cut towards this end or it may cut towards this end. So, and then we have certain proteins, DNA helicase enzyme, exonuclease 1 or exonuclease X10 or exonuclease 7 or RECJ nuclease in this case. Again, ATP dependence. So here a whole chunk of DNA is removed. So either in this, in any of these, you can see, and then DNA polymerase 3 along with single strand binding proteins that completes this strand in both, whether this or this one. So overall, as you have seen that this mismatch is recognized by these proteins, and there is a cut in the daughter strand on any of those sites, and then whole chunk of DNA is removed and followed by DNA polymerase 3. In this way, this particular mismatch is repaired. Now come to the second part, which is base Excision repair. Here different proteins are, again you can see these are used, and base Excision means there is an abnormal base due to certain chemical or physical mutagen, for example. So if uracil is there in case of DNA, uh, DNA contains thymine. So but if uracil comes in, or hypoxanthine, xanthine, or other alkylated bases, and also in some cases pyrimidine dimers. So in any of these cases, what uh, system is which uh, helps in the repair is the base Excision repair system. So this is the damaged base, In here at this place there is a damaged base. This damaged base is removed with the help of DNA glycosylase. And then another enzyme, AP endonuclease, it creates a nick at this place. So this is the second step. And then third step is DNA polymerase 1 enzyme. It performs this function, the special function, 5' to 3' exonuclease activity, just like what it does during removal and replacing of the RNA primers during replication. So DNA polymerase 1 replaces some of the nucleotides, and it's this nick basically is a place where this DNA polymerase 1 can start its function. And finally, DNA ligase, it seals this nick as in the DNA replication. So in this way, uh, if some, this is called as base excision repair, if some base, some uh, change takes place in any of the nitrogenous bases, that is recognized by DNA glycosylase, followed by AP endonuclease, then DNA polymerase 1, and followed by DNA ligase. So here you can see more clear picture of these. So now comes the third, which is nucleotide excision repair, and you can see how many different ABC exonuclease, DNA polymerase 1, and DNA ligase. And this nucleotide excision repair is required for larger DNA structural changes like DNA lesions when they are produced. So here you can see this is a DNA lesion in the red in a certain part, and comparison is given with between the E. coli system and the human system, human cells, how this repair takes place. In case of a lesion, there is an enzyme which is exonuclease enzyme, and this enzyme uh, makes two cuts on two sides of the lesion. As you can see in case of humans, you can see here and here, and in case of E. coli, you can see here and here. This then DNA helicase in both of these, it removes these DNA which contains cut on both ends. It is 13 mer, meaning 30 nucleotides long in case of E. coli, and 29 mer, meaning 29 nucleotides long in case of human cells. So you see that this part is removed. So DNA polymerase 1 in case of E. coli and DNA polymerase epsilon in case of human cells, it regenerates this uh, part of the DNA which is removed, and followed by sealing of the nick with the help of DNA ligase in any of these. So this is nucleotide excision repair when damage is in the form of a DNA lesion in certain region. Now comes the fourth type, which is direct repair. Here there are enzymes which directly takes place without the system, without any set of enzymes. So they can make this change and repair this DNA. And you may see that pyrimidine dimers are present here, this example of this, and in this case direct repair as well. So we take examples one by one. This is the pyrimidine example of the pyrimidine dimers. Pyrimidine dimer means when two pyrimidines side by side, for example two thymine are there is a covalent linkage between the two uh, pyrimidines which are in the same strand side by side. Usually there is no such type of linkage that is there. So if this linkage, such type of linkage is seen in case of in terms of pyrimidine dimers, in the next round of the replication, what will happen that DNA polymerase will think it of as something else, so that will contribute to mutation in the DNA in the next generation. So it must be removed. So I'm not going into much of the chemistry here, but this is the overall picture of this pyrimidine dimers. In some cases, there is nitrogenous bases are methylated, like for example O6 methyl guanine is example here, and the enzyme methyltransferase enzyme that simply works here and removes this methyl group, and you see the guanine nucleotide once again. Similarly, in case of uh, so here you can see the example of where the if methylation is there, like in this O6 methyl guanine, this guanine recognizes thymine. This is a big change in the DNA that may take place. Normally, guanine pairs with the cytosine, but if the methyl group is there in the guanine, it will recognize thymine. So it means when this DNA replication will take place, this O6 methyl guanine will introduce thymine in the daughter strand rather than cytosine. So it means there will be GT, and in the next round of replication this T will accommodate A. So it means it is altogether changed from GC to AT. Here it is, you can see elaborated. This is GC, and with due to this methylation, this G will recognize T in the next round of replication, and in the next round, further round of replication, this T will recognize A. So you can see it, we started with GC at first place and we ended up with AT. So it may be lethal for the cell or some mutation that may some harmful effect that may create like total altogether change. And remember, if the mutation is within the place where within the gene which corresponds to some active part of the protein, such as active site of the enzyme or important part of some proteins like transport proteins trans or something, so that will have a significant effect. But if the mutation is in some inactive part of the gene or outside the gene, it may be just a silent mutation having no effect. These are some other examples of ALKB, and you can see 1-methyl adenine. It has similar effect as for O6. So here this is has to be removed. So similarly, 3-methyl cytosine, methylation in cytosine is removed by some enzyme. And another enzyme which I haven't incorporated here is that directly removes the pyrimidine dimers. And that enzyme is photolyase, as its name implies, because this pyrimidine dimers, they may be formed by the due to the UV light, due to the electronic transitions, and then photolyase is the enzyme that may remove those particular the linkage between the two pyrimidines. In some cases, there is a recombinational repair system that. So if there is an unrepaired lesion, then there is a single strand DNA or double strand DNA break in, and there is a repair which is called as recombinational DNA repair. And but this recombinational DNA repair is error prone repair. It means DNA, the cell is trying to save its DNA as much as possible, but it contains some errors in it. Then in case of severe stress on the cell, uh, for example, there is continuous ultraviolet radiation or something, in that case, a big set of machinery that is activated. And this is called as SOS response, signal of stress response. And here, multiple genes are induced all at once. and these genes correspond to certain proteins. As you can see, many of these proteins like DNA polymerase 5, ABC, XC nuclease, similarly Rec A protein, and DNA polymerase 4. All these different proteins, they are required for the to save the DNA damage. And there are certain genes involved in DNA metabolism, but role in DNA repair is still not known. They are also activated, and genes of some unknown functions. So meaning is whole set of genes are activated in SOS response, and usually these are activated due to a certain protein which is called as Lex A. Lex A is a protein, and that protein, Lex A protein, is basically a repressor protein, Lex A. And this repressor protein, as its name implies, is a repressor protein, meaning it represses all these different genes which are required for the signal of stress. So this protein represses, though normally these genes are not activated. But as soon as some signal of stress comes, like a cell is under severe damage situation, in that case this protein is degraded. This protein is degraded by uh, by an enzyme which is by a protein which is Rec A. This Rec A protein, basically, this Rec A protein, it degrades this Rec A protein. It degrades this Lex A repressor. So because this repressor is repressing all these genes, so as soon as this is degraded, you see all these genes are activated. So there are in many more types of, you can say, repair systems that exist in E. coli, but I just have given you an overview of some of the very important ones, so give you an idea, and you can also have an idea that how these systems work in different types of cells. And of course system is more complicated in eukaryotes, uh, but I end my lecture here as per requirement of this particular course.